Hi, my name is Leah Kaplan, and I'm a research associate in Aviv Regev's lab of the Broad Institute. We are interested in which factors contribute to cancer progression, specifically looking at gene expression of individual cells and in their original context within the tumor. We think that tumors are composed of different microenvironments that are characterized by these genetic and spatial factors. Tumor microenvironment is basically like a little neighborhood that is characterized by the different cells that are present and how they're behaving and the different molecules that are present there. Another way to understand the importance of the spatial positioning of these cells within the tumor and the tumor microenvironments is you can think of a single cell in a tumor like a person in a country. And if she were to travel to a different country, she would be interacting with different people or different cells. She would be exposed to different things, new foods, a different culture, even different weather, different climate. And due to all these factors, she would probably behave differently. And this is similar to how cells in different environments will behave differently. So this is why it's important to understand a cell's positioning and how it influences behavior to better understand cancer progression and to better understand cancer prognosis. The goal of the project is to understand the interaction between cancer cells in the context of their tumor microenvironment. There are many different types of cells that make up a tumor besides just the cancerous cells. And these cells include cells of the blood vessels and immune cells and other cells that supported the functionality of what was the normal and healthy tissue. Understanding the spatial position of these cells and which cells are interacting with which, going back, thinking to which neighbors are interacting with which within a neighborhood, can help you understand the cell behavior. The image you see is of a resected colon tumor from a mouse that was modeling human cancer, and this mouse was generated by the Yilmaz lab. The green fluorescent labeling is labeling individual cells within this tumor. And because everything you see is tumor, all of these green cells are cancerous. The blue inside of these cells is labeling the genetic material. The yellow is labeling stem cells of the colon. And the red is labeling cells that are actively proliferating. So where you see the yellow and red labels together, you see actively proliferating stem cells. And because you're looking at a tumor, these are actively proliferating stem cells of a tumor. And that is very interesting to see that these are not homogeneous throughout the entire tumor. It is not spread evenly throughout the entire tumor, but rather these pockets of proliferating stem cells are localized to specific regions of the tumor. So this is maybe a neighborhood of proliferating cancer stem cells. And this was not exactly what we expected to find. But it's interesting to see that there are specific regions, and then that leads us to ask, so what's different about these different microenvironments versus the rest of the tumor? Which cells are interacting? What molecules are present? What are they being exposed to that causes them to act differently in this region of the tumor relative to other parts or other neighborhoods? Cancer affects so many people. Multiple loved ones of mine have passed away due to cancer, and I've always had a drive to just understand how things work and why things work the way they do. And being able to combine these genetic and imaging technologies, I think, really gets to the bottom of that in terms of solid tumors, of understanding really how do these factors, these genetic and spatial factors, come together at the single cell level and then cause tumor progression. And just by being able to work in a field that I know will affect so many people out there, I really enjoy.
What gets you excited about this project?
Science is cool.